Hi guys, I'm one of your SI leaders, Natalie Michelle for BIOL 005A. Let's start talking about this video. So the summary of this video is going to be cellular respiration and a little bit of photosynthesis. This is based off Mondays and Wednesdays lectures, so let's get into it. So remember the equation for cellular respiration is glucose plus oxygen makes uh, carbon dioxide, and it makes water, and it makes ATP. So the way I like to memorize it is that glucose is a sugar, which is sugar's food. Oxygen is what we breathe in because we do cellular respiration. Uh, Carbon dioxide is what we breathe out when you just, you know. And then water, you can think of pee or sweat, doesn't matter. It just helps me think of that. And then ATP is the energy, hence why we eat food. We eat food to get energy. So make sure you memorize that equation. So cellular respiration goes in 4 phases, right? We have stage 1, glycolysis, stage 2, pyruvate oxidation, stage 3, the citric acid cycle, also known as the Krebs cycle, and then stage 4, which is oxidative phosphorylation. So if you can see here in this step right here, glycolysis is the only one that's not in the mitochondria. Remember that. So there's some like bacteria or single-cell organisms that do not have a mitochondria. Mitochondria is more of like a complex organelle, hence the double membrane. That's how you can kind of tell something's more complex organelle. I'm not going to go more detail about that. So glycolysis you see is in the cytoplasm, cytosol, and simple organisms that don't have mitochondria do glycolysis for their energy because they get the 2 ATP net. So some tiny molecules don't need it. So that's why that's out of the mitochondria. So remember that. Everything else from there on needs oxygen to get into the mitochondria. Like glycolysis doesn't need oxygen, so no needed, right? Needed, sorry about that. But everything else does need oxygen. Hence the word oxidation, oxygen, and oxidative phosphorylation. Citric acid cycle also needs oxygen. You need oxygen for pyruvate oxidation. Therefore, the next step, you need oxygen in the first place to get there. So yeah, okay. And remember, oxidative phosphorylation has 2 steps. It has the electron transport chain and it has chemosis. So remember that. It's still part of the 4th stage. There's just 2 steps into that. So let's talk about the summary of like the products that are happening in cellular respiration. So we have glycolysis, pyruvate oxidation, citric acid cycle, and the ETC/chemosis. So one glucose molecule makes about 32 ATP, right? So you got to remember we're not talking about one pyruvate, we're talking about one glucose. So that means in glycolysis, it makes the 2 pyruvate molecules. Then you go over pyruvate oxidation twice, and you go to the citric acid cycle twice and stuff like that. Remember that. So it's already counted for here that we go through pyruvate oxidation twice here, citric acid cycle twice. This is all counted for one glucose molecule. But just remember, just be careful on exams. You have to read if it's doing one glucose molecule or is it talking about the one pyruvate. So glycolysis, you make 2 ATPs and 2 NADHs. In pyruvate oxidation, you make 2 CO2s and 2 NADHs. Citric acid cycle, you make 4 CO2s, 6 NADHs, 2 FDH2s, and 2 ATPs. In the electron transport chain and chemosis, you convert those electron carriers, which are the NAD, H, and the FDH2s into ATP. We'll talk a little bit more about that in detail. And then this is where you use the water, the oxygen to make water. So remember, oxygen is the final electron acceptor, and that's why you need oxygen because it's like an assembly line. If you don't have oxygen at the very end picking up the hydrogens, it's going to back everything up and it's not going to happen. So let's talk about a little bit about the electron carriers and what it is to convert into like ATP. So think of ATP as cash. It's energy, they can use it right away. Like when glycolysis, the 2 ATP, it's just going to go out there and be used for whatever it needs. Then you can think of electron carriers, which is NADH and FDH2 as checks. You need to convert your checks into cash before you can use them. You can't just straight up use a check. So, from what the past I learned that NADH equals about 2.5 ATP and FDH2 equals 1.5 ATP. So from this, if you calculate, I'm not going to do the calculations, from all these NADHs and FDH2s, you get about that 28 ATP. And then the 32 is the 4 ATPs that you get over here. So 28 plus 4 is 32. So just remember that, so they also have different calculations. Like if you actually look at this photo right here, it actually says 36 ATP. So it's varied because NADH does not make exactly 2.5 and FDH2 does not make exactly 1.5. It depends on how they sum it up. So maybe some textbooks can say NADH makes 2.7, FDH2 makes almost about 2. You know, it just depends on textbooks and all that. So just rule of thumb on that. So yeah, that is cellular respiration in a summary. If you guys have any questions about cellular respiration, make sure you go to office hours, go to an SI session. You can even comment on this YouTube video. We are going to answer any questions that you guys have. So yeah, if you have any more questions, let you go. That is a summary of cellular respiration. Okay, so now we're going to talk about anaerobic respiration. So this is basically if you don't have oxygen, so you can't do pyruvate oxidation and BIOL. So this is just glycolysis. You're just able to do glycolysis right here. So glycolysis only to make your ATP, but there's nothing else. So this is anaerobic stuff, right? So anaerobic means no oxygen. I think of the N in anaerobic as no oxygen. So I think of the N for no. Okay, so like I said earlier, no oxygen as final electron acceptor, right? So in glycolysis, you make this NADH. And the NADH is used for the electron transport chain. But if you don't have oxygen, you're not doing the electron transport chain, right? So the point of fermentation, which is what we're talking about, is the idea of no oxygen and glycolysis happen, and you have this NADH. So if you're a little bacteria and you only use glycolysis, you need to keep making this NADH to NAD plus so you can keep doing glycolysis, because in the step you need to convert NAD plus to NADH, right? So to continue glycolysis, you can't just have NADH sitting in your cell for no reason, right? You need to replenish that so you can keep using it and recycling it, right? So that's what fermentation is. It's it needs to be recycled or like reconverted to NAD plus, oxidized. If you guys remember that, it's going to be oxidized again. So this allows glycolysis to continue, right? So you need to recycle that NADH to NAD plus. There's two types of fermentation. There's lactic acid fermentation, which is done by animals like us. I think of lactic acid as like lactate, like the milk, and you know animals, well, technically mammals do that, but if that's a way to help you memorize it, that's how I thought of it. And ethanol fermentation is actually how alcohol, like beer, that type of stuff is made. And if you guys know, beer is made from yeast and all that type of stuff. So if we can't do ethanol fermentation, otherwise we would just be randomly drunk, you know, if we're not having oxygen in our muscles or something like that. So just remember that. I don't have that on the slide. Just remember lactic acid fermentation, animals, and ethanol fermentation is bacteria and yeast. Okay, so now let's go into photosynthesis. So in photosynthesis, the equation is actually the opposite, if you notice, from cellular respiration. We have our CO2, our water, our light, except for light's different, and our glucose and our oxygen. So if you remember this, your plants need CO2. They say plant more trees with photosynthesis to help, you know, plants to reduce the carbon dioxide in the environment, stuff like that. You know, you have to water your plants and you need to give your plants sunlight, right? That's how you basically take care of a plant. And in return, your plant will give you food, glucose, sugar, stuff like that, and oxygen. Your plant makes oxygen. So that is the summary of the formula. It's just the opposite. If you remember, cellular respiration is an exergonic reaction, while photosynthesis is an intergonic reaction. They're opposites, and their equations are opposites, so it makes sense. So it's just saying that it converts, it uses sunlight, which is photon energy, to make the CO2 and the H2O into oxygen and glucose, right? So photosynthesis has two stages. So it has the light reactions, which is the steps that need the light from the sunlight, and the Calvin cycle that actually doesn't need the light. So it's also called the dark reaction, but it does not only happen at night. It can happen at night, but also happens in the daytime. It's just the one that doesn't need light. So light reactions actually occur in the thylakoid membranes, which is this thing right here. I'm going to also do it in red. Is this green thing. This is where the thylakoid, and it occurs in its membranes. It's also a double membrane molecule in a double membrane thing. Yeah, weird. So light reactions, it occurs in the thylakoid membranes, and this is where the solar energy is absorbed, right? This is where the sunlight happens. This is where it requires water. This is where water is required. And this is where it produces oxygen from the water, actually. Energy is stored in electron carriers. So we have another electron carrier that we have to learn, and also ATP. So this electron carrier, if you notice, this one has a P. You can think of the P for photosynthesis. It actually stands for phosphate, like one of your functional groups, if you remember. Stands for P for photosynthesis is just a way to memorize it, but it's actually phosphate. So remember that NADPH is for photosynthesis and NADH is for cellular respiration. So now let's talk about the Calvin cycle. The Calvin cycle occurs, let me just do this one in a different color. The Calvin cycle occurs in the stroma. Okay, the stroma is kind of like the cytoplasm area, see here? It's kind of on the outside in the blue cytoplasm-ish type of area, which is called the stroma. So don't call it the cytoplasm, call it the stroma. This is where it requires the carbon dioxide, and this is where the glucose is produced. And it uses the energy from the NADPH and the ATP to produce this stuff. So think of light goes first, and then it becomes the Calvin cycle. Light first, then Calvin, right? So that is a summary of the two stages. There's more details on it. On the next slide, I'm going to talk a little bit about the light reaction. This is a photo from Khan Academy. So if you see the chloroplast, and if you see they're zooming into the thylakoid, and this is where this box is. So I'm going to just zoom in a little bit on here. So in the light reaction, we have these photosystems, photosystems 2 and photosystem 1. Photosystem 2 is first. Scientists accidentally discovered the first photosystem, which is technically the second photosystem, first. And then they're like, oh, shucks. They found another photosystem, and they found out that that one was first. And, you know, it happens. They make mistakes. So it's a little bit screwed up. And don't let it confuse you, though. Okay, so let's talk about photosystem 2 first. So if you see here, it's in the stroma on this side, and the other side you'll see on the other corner. It's the thylakoid lumen, so the interior of the thylakoid, right? So if you see here, we have our light. It hits the photosystem, and it hits at the rate at 680. That is just the absorption rate, okay? Don't worry too much about that. So what we see here, I'm going to kind of draw the path of electrons in blue. So water gets split into oxygen and hydrogen, and the electron goes into the photosystem. It gets all excited by the light. The light like basically dings it, and it gets really excited, and then it goes down this electron transport chain. Wow, sounds similar to cellular respiration. And as you see, we see protons going down, or going up its concentration gradient. It's actually going against, it's going from low to high. So this is high hydrogens, and this is low number of hydrogens. Remember, it takes energy to go against the gradient. So more hydrogens are being put on this side, right? So that looks very similar to cellular respiration, right? So now the electron, after it goes down the gradient, just think of it getting less and less excited as it's going down the blue arrow. And now we have to excite it again, right? So light comes in again. This time it comes at a rate of 700, excites the electron again, and this electron goes down a gradient again, not as crazy, and the electrons get picked up by NADPH, and you can think of the electrons as in this hydrogen molecule right here, makes NADPH, right? Very similar to cellular respiration, if you see here, right? Except it's usually the H gets dropped off from the electron carrier, but we're picking it up, right? So now we're doing kind of like what chemosis did. So we have a concentration of high hydrogens at the ATP synthase. So the hydrogens will go towards equilibrium and they will go here, and it'll do the same thing as in cellular respiration. It makes ATP, it makes energy, right? That's all that is. That's the summary of the light reaction. And that's how your electron carrier is made here. And this is how your ATP is made. And then this stuff will go into the Kelvin cycle. I'm not going to be covering it in this video. And then glucose and all that's produced. So I hope that was really helpful for you guys. Good luck. Study well. Yeah, and I will see you guys in the future. Bye, guys.